The overall goal of this procedure is to demonstrate a method to separate neutrophils from a drop of whole blood and then perform a subsequent chemotaxis assay using a single microfluidic device.
This method provides a detailed protocol to perform neutrophil chemotaxis assay using small volume of whole blood, and can be easily applied by researchers to answering biological questions related to immune cell migration. The main advantage of this technique is the integration of neutrophil isolation and a chemotaxis assay on a single microfluidic device. This permits rapid sample-to-result analysis in 25 minutes.
To begin, prepare the master mold as described in the accompanying text protocol. Then, silinize the surface of the SU8 mold to facilitate the release of PDMS from the mold. Next, mix 40 grams of PDMS base and 4 grams of the curing agent in a plastic beaker. Place the prepared SU8 master mold in a petri dish and carefully pour the mixed PDMS solution onto the mold. Place the petri dish in a desiccator and apply vacuum to degas the PDMS solution for 20 minutes. Then, place the petri dish in an oven and cure the PDMS at 80 degrees Celsius for 2 hours. After baking, carefully cut and peel off the PDMS slab from the SU8 mold. Cut the PDMS slab to separate it into individual devices. Trim the PDMS device to make 2 walls around the cell loading port for holding the magnets. Next, punch out the cell loading port using a 3 mm diameter puncher. Then, switch to a 6 mm punch and remove the chemical inlet reservoirs in the waste outlet.